Hello, thank you very much for joining this webinar titled Microfluidizer, a potential tool for process development to make nanotetrac for pharmaceutical applications. My name is Stephen Masidi. I'm the Director of Inside Sales and Applications at Microfluidics. And with me is Dr. Dhruva Barali, who is the Assistant Director of Nanotechnology at Albany College of Pharmacy and Health Sciences. Dr. Barali and I have been working on these nanotetrac particles, they're polymer nanoparticles, for a few years now. We have established a collaboration to perform process development of these particles, but to be fair, Dr. Barali has done most of the work. His background is in chemistry. Specifically, he has a PhD from the University of Delhi in India in nanotechnology and drug delivery. So to start, I'm just going to give a quick overview of Microfluidics, the company, and the products and technology we offer. It will be quick, probably about 5 minutes. And then after that, I'll turn it over to Dr. Barali, who will give the bulk of the presentation and talk about his process and his research. So Microfluidics has been around for about 35 years, producing these high shear fluid processors based on our patented interaction chamber technology. We're headquartered in Westwood, Massachusetts, which is just outside of Boston, and we have localized sales support in almost 50 countries worldwide. And over the past 35 years, we've sold over 4,000 microfluidizer processors to 2,000 different companies. Microfluidics was acquired by IDEX Corporation and grouped with Quadro Engineering, Patrick Corporation, and MatCon to form the Material Processing Technologies platform. Microfluidizer processors are used for both research and development and for manufacturing of pharmaceutical ingredients, vaccines, inkjettings, coatings, nutraceuticals, and cosmetics. Microfluidics has vast applications and machine design experience, so no matter what nanotechnology application you are currently working on, odds are we can help accelerate your process. Because really our goal is to enable our customer success. So this is a little bit about what we do best. Our technology is best for generating nanoemulsions. We also do a lot of work with cell disruption for biotech applications, for protein production. We've done a lot of research about polymer nanoparticles, which Dr. Barali is going to talk very specifically about. And we've also done quite a bit of work with porous polymer microspheres. So if you have any questions about that, let us know and we can provide you more information about those. And we also do a lot of work with liposomes, particle size reduction of various solids, pharmaceuticals, and other chemicals. We do deagglomeration of things like graphene, carbon nanotubes, cellulose, things like that. And our technology can also be used for molecular weight reduction in certain applications. And then we have a line of production machines where it's completely linearly scalable from the lab scale to the production machine. So our technology can produce the exact same results when you're doing your R&D as when you go to scale up, which is very important for most applications. This is a schematic of our technology. Essentially, there's an inlet reservoir where you feed your material. And then our technology requires the use of a constant pressure intensifier pump. And then there's our fixed geometry interaction chamber, and that's where the high shear and high impact occurs. And I'll talk about that a little bit more in a minute. And then there's a heat exchanger or a cooling coil after that to cool the material after processing. All of our pumps are custom built to accommodate very difficult materials. We can process materials with high solid contents and high viscosities. We can work with a wide range of temperatures as well. Also, cooling occurs quickly so that we can process very temperature sensitive materials like proteins. So here are two schematics of our interaction chambers. We have two types. The one on the left is the Y-type interaction chamber. In this chamber, the material enters through the top. It is split into two streams, and then those two streams are forced to impinge upon each other in the high shear zone. It's usually used for mixing applications like emulsions and liposomes and things like that. The one on the right is the Z-type interaction chamber. It's typically used for particle breaking, like um, dispersions and cell disruption. They're very resistant to abrasive forces because they're manufactured out of polycrystalline diamond, typically, and they are very easy to clean because there's virtually no hold-up volume inside the interaction chamber. The other benefit of these interaction chambers is that since they're so small, typically in the range of about 100 microns, we can array them in parallel for scale up. So you do your developments with a single microchannel, and then when you go to scale up, we just use a larger pump and align multiple microchannels in parallel, and that way you get the exact same velocities, the exact same shear rates, the exact same energy dissipations on the production scale, and virtually always we're able to generate the same particle size distribution on the production scale that we were able to generate on the lab scale. So as a summary, microfluidizer technology uses constant pressure processing with very high potential processors, and we have fixed geometry interaction chambers with multi-slotted interaction chambers for scale-up, which means that you can generate very small particles with very consistent processing, very narrow particle size distributions, and very good batch-to-batch repeatability, as well as guaranteed scale-up from the lab scale to the production scale. And this is just a brief glance at our product portfolio. We have a wide range of processors with different volumes, different energy input requirements, and different levels of cleanability with a lot of cGMP options. So without any further ado, I'm going to turn things over to Dr. Barali. Thank you very much. And here's Dr. Barali.
Okay, so thank you very much, Steve, for your introduction. And from here, I will take. So today I'm going to talk about a little bit about what we have used microfluidizer for in our lab. We have developed a method and which was in a laboratory setting, but we have lots of trouble for scaling up. So I will give you a little bit introduction what we have done, what was the problem, on how microfluidizer helped us for scaling up the method. So this is my outline of the presentation. So I'll give you a little bit introduction about the nanotechnology. I'm pretty sure most of the audience know what is nanotechnology and nanomedicine, but I was just gonna give you a little bit introduction from my point of view. And nanoconjugated inhibitor, angiogenesis inhibitor, what we have been working with, how to scale up this nano formulation from laboratory setup, then in vitro efficacy study from this formulation, in vivo efficacy study in different kind of mice model, and finally a little bit summary that which everybody actually give a little bit summary of the presentation. So as everybody knows, like what is nano, I'm not going to explain about the nano, but why nano? So what is the advantage of nano in medicine? So what we used in our lab is a chemotherapeutic drug, and most of the chemotherapeutic drugs are not soluble in water or any kind of hydrophilic solvent. So nanoparticles can increase the solubility of the drug, water insoluble drug. At the same time, it can increase the stability of the drugs, like many proteins may not have that much of stability while it is in a hydrophilic solution. So nanoparticle, by masking the protein or other kind of unstable molecule, it can give the stability for the formulation. Specificity, meaning it can be used as a targeted delivery. The nanoparticles can be used as a targeted delivery by putting different kind of targeted moiety on the surface of the nanoparticles, not a single, even multiple kind of targeting moiety on the surface of the nanoparticle. We can put a imaging probe to track the nanoparticles. We can put different kind of anti-cancer drug inside or in the surface of the nanoparticle depending on the use. And the surface coating, meaning we can actually coat the nanoparticles depending on what kind of nanoparticle and where actually you want to go the nanoparticles. For example, PEG, polyethylene glycol, or other kind of surface coating we can use for the nanoparticles to give more stability and more targeted delivery. So by saying that, we can increase the specificity and the multimodality, meaning targeted moiety, imaging probe, anti-cancer drug, surface coating all together. And the efficacy, while doing that, we can increase the efficacy of the drug. So our concept, so we actually took a drug, that's a pretty common, well, it's not even a drug, a common moiety, which is a thyroid hormone. So it's L3. So people have seen, or there are lots of clinical trials where it has shown that it can increase the survival rate in breast cancer, lung, glioblastoma, and many others. This is the major advantage of using a thyroid hormone, say L3, Tetrac, and many other thyroid hormone. But one of the major disadvantages of this drug is it can exert unwanted genomic effect. So it's a hormone, so it always has a tendency to exert genomic effect. So what we wanted to do, we just want to eliminate the unwanted genomic effect. At the same time, we want to exploit the anti-cancer uh, efficacy of this hormone. So we thought, okay, we can actually do that by conjugating to a nanoparticles, so that it, because of the size, it will restrict it to go by going to the nucleus. At the same time, we can actually get the anti-cancer efficacy of the drug. So just an example of the proof of the concept, what we did is we conjugate thyroid hormone with a dye, LXA4488 in this case, just without anything, just the hormone to conjugate it to a dye. And in the next picture, what we have done is a nanoparticle conjugated to the thyroid hormone. At the same time, a dye is also conjugated to the thyroid hormone. And you can see the dye itself, what it is conjugated to the thyroid hormone, it can go to the nucleus, but when it is conjugated to the nanoparticles, it cannot go to the nucleus. So this is the proof of the concept that we can actually we can prohibit the nanoparticles going to the nucleus while carrying a thyroid hormone on it. That's what exactly we wanted to do. So this is basically a very common flow diagram how to make nanoparticles. So we have used a very common polymer, which is biodegradable polymer, PLGA, polylactide co-glycolide, and we have a surfactant, polyvinyl alcohol. So you can see it's a simply a diffusion method to make the nanoparticles, where you do the dialysis and then you can lyophilize it to get a powdered form. So next step was, well, this is just a conjugation method. It's a very simple chemistry involved, so I'll not discuss it in details. So you can see the structure here. So finally we can get our nano formulation conjugated to the thyroid hormone, which is tetra in this case. So we have determined, or we have characterized the nanoparticles with dynamic light scattering, transmission electron microscope, and you can see here the size we can restrict in between. Well, we can manipulate the size, but what was favorable size for us was around 200 nanometer or less than 200 nanometer. So both was actually confirmed by dynamic light scattering and the transmission electron microscope. So we have actually published at least 15 different original research articles showing that our nanoparticles can inhibit the growth of non-small cell lung carcinoma, follicular thyroid cell carcinoma, or many others. Even it can actually inhibit the growth of drug-resistant breast cancer cell in mostly in the mice model. But the question comes, what next? So now what? So how to scale up this nanoformulation for preclinical study? So let's synthesize 100-gram tetrac equivalent of the formulation. So what we started here is 10 ml in the lab, so which give us 0.15 mg tetrac equivalent. Then when you go to half a liter, it gives us 7.5 milligrams of the tetrac equivalent in a nano formulation. And for 150 milligrams of the tetrac equivalent, which is the API, we need 10 liters of the liquid. So we calculated, okay, we want to synthesize 100 grams of tetrac equivalent, then we will need 6,666 liter of the liquid. So that's where the problem starts. So where to get such kind of like a reservoir, maybe a King Kong reservoir or something like that. So that's kind of a very, it looks like it's a very absurd idea to scale up this formulation. So we went back, okay, we have already worked hard, now it's time to work a little more smarter. Okay, so what do next? So we read some literature and make few calls and saw the Microfluidics. This company is just in our neighboring states, which is a 2-hour drive. So we call Brian, the person who actually is the in charge for selling this in the Northeast, and he actually guided us what we need. So he said, okay, we have a small machine and which can be used what we are looking for. So he said that they can give us like a one-day training in their facility and see whether you can make the nanoparticle using this machine. And this was like LM10. So we came to Boston with our chemicals and try to make some formulation. So I'm just giving you an idea how you can make our nanoparticles, and we called it NDAT nowadays. This is the standard nomenclature I'm going to use in the rest of my presentation. So basically you can mix some ethyl acetate, PLGA, and NAT8, which is our tetra conjugated PLGA nanoparticles, tetra conjugated PLGA polymers. Then we do the homogenization with either magnetic stir or a homogenizer. Then we can use this machine LM10, and at 20,000 psi we can make those nanoparticles, which will be a size around 120 nanometer to 150 nanometer, and you can actually remove the solvent either by dialysis or other TTF, and you can lyophilize it to get the PLGA tetrac nanoparticles in powdered form. So when we calculated back, it turns out that we don't need 6,666 liter of the solution or the liquid. Now with this microfluidizer, we only need 85 liter of the liquid to make the same amount of tetrac equivalent conjugated to the nanoparticles. Yeah, now we can say, okay, now we got a method. So that's the machine actually we can use for scaling up. Then we made the nanoparticles with this microfluidizer LM10 in a, say, 1 liter amount. Sometimes we can even make around 50 ml of the nanoparticles. So this machine is pretty much flexible, making nanoformulation in the laboratory settings. So I myself make around 50 ml nanoparticles. That's the smallest amount generally I make, but I also make about 5 liters of the nano formulation in the machines. Well, you need a little bit of patience, but it's worth it. So all those nanoparticles, which is NDAT or nanotetrac, we have actually analyzed by using UV spectrophotometer to see how much tetrac equivalent is conjugated or in the nanoparticles. The size was analyzed by transmission electron microscopy. We also check out the cellular efficacy, in vivo efficacy in different kind of tumor model in mice, then IVIS imaging, which give us the conjugated dye-conjugated nanoparticles where it is going in a live animal, and the amount of tetrac or other kind of drug in the nanoparticles can be measured by LC-MS. So those are the different kind of characterization technique we used after synthesizing those nanoparticles in the microfluidizer. Just give you an idea about the size of the nanoparticles by dynamic light scattering. It was around 144 nanometers, and it can actually go down to 120, and even some cases depending on the different kind of parameters, it can go up to 100 nanometer in size, and you can get a very monodispersed nanoparticles. The polydispersity factor was 0.046. It's almost equivalent to a standard that you can use in dynamic light scattering. And transmission electron microscope also support our fact that nanoparticles are around 140 nanometer in size. So as I mentioned before, so my first trip was to Boston to Microfluidics facility was the proof of the concept whether we can make nanoparticles with their machine, and answer is yes, we can make the nanoparticles with LM10 microfluidizer, which is the smallest version of the microfluidizer available that time. Then okay, we can make nanoparticles say up to 5 liters, so but still it's not enough for the commercial manufacturing. So what I did to check, okay, we scale up the nanoparticles to certain extent, so is there anything else, like is there any can we go further in terms of volume by using their other kind of machine or microfluidizer in the facility. So second time I make the trip just to make sure that whether this scale up nanoparticles can be further scaled up in by using other kind of machine or microfluidizer in their facility. Well, you can see here, so microfluidics people, particularly Steve and Kristen, they are in work. So we are making nanoparticles, say liters, using a different kind of model, which is M1110EH30K. And interestingly, the size of the nanoparticles we made in LM10 and size of the nanoparticles we made in M1110EH was almost, well, if not identical, it's more or less similar. So whatever we was looking for, we get the same size nanoparticles but in a different amount of fluid. So this is the proof that we can actually use different kind of machines, but still we can get the same kind of nanoparticles we are looking for. The different parameters that affect the synthesis of our nanotetrac is the number of pass, meaning how much passes we have used in the instrument, the pressure, and the type of chambers what we have been using in the microfluidizer. So we have done a lots of permutation combination, and we figured it out that our nanopart formulations, that what we are looking for, can be synthesized using a chamber which is F20 chamber with gap spacing 75 micron, and the pressure we are using was 20,000 psi, and the number of pass was 5. Well, we can actually shorten this pass to 3, but the number of pass 5 gives us the best and reproducible result. So we used those parameters for the synthesis of our nanoformulation in rest of our formulations. And this is actually this is an example. Actually, I should actually present this slide before. So how actually different kind of instrument you can use in the different kind of instrument or microfluidizer you can use without affecting the size of the nanoformulations. So we have used, say, 1 gram of starting material in 30 ml, and we get 150 nanometer size nanoparticle in LM10 microfluidizer. And at the same time, if we use 100 grams of our starting material in 3 liters, and even if we use either LM10 or M1110EH microfluidizer, still we get the same kind of nanoparticles what we are looking for. So I would say, or my recommendation will be, the size actually you can get exactly same size nanoparticle using different kind of microfluidizer, and even the different amount of volume or material you are using. So it's not a factor in this case for synthesizing the nanoparticles. Well, the good news about our nanoformulation is that it get orphan drug designation for the following cancers: is glioblastoma and pancreatic cancer. So meaning we can have an access to a very small amount of patients where we can test our formulation in hospitals, and we already have a IRB for glioblastoma and pancreatic cancer in MD Anderson. Now once we make those nanoparticles using this instrument, we actually checked whether these nanoparticles has the similar kind of efficacy in vitro and in vivo, and we are checking it whether the nanoparticles actually can go or can be restrained from going to the nucleus. So for that, actually we used U87 cell to check the uptake, since we are using our nanoformulation for glioblastoma. And we have actually labeled the nanotetrac synthesized by microfluidizer by Cy5 dye and checked the uptake uptake by confocal imaging. So here you can see the nanotetrac labeled with Cy5 doesn't go inside the nucleus in U87 cell, but when the tetrac was labeled with Cy5, it is all over the cell, meaning it can actually penetrate to the nucleus. So our nanoparticles can block tetrac from entering to the nucleus, which was our primary objective of the formulations, even if we use the microfluidizer. The next step was we checked the efficacy of our formulation in glioblastoma xenograft, and you can see here after 10 days there is a tremendous inhibition of the tumor volume when we use nanotetrac 1 mg per kg, while the control doesn't have any effect. So it's keep growing, the cancer keep growing in the control while in nanotetrac you can see there is a tremendous inhibition of the growth of the tumor. Tumor weight also same thing. And next slide is another kind of cell line which was U87 and luciferase expressed. So you can see control has where the luciferase, we can actually image the luciferase using IVIS imaging. Control has a lots of luminescence coming out, white PLGA nanoparticles, lots of luminescence coming out, meaning the cancers are growing. But nanotetrac you can see in many cases they are totally gone, or in some cases they are diminishing, meaning this nano formulation can again kill the cancer cells in the tumor. Similarly, we actually check the histopathology of the tumor. We cut the tumor and make slice and check it out, and you can see a complete necrosis of the formulation of the cancer cell when you use nanotetrac 1 mg per kg. So those are the different kind of like factor which actually affect cancer, like apoptosis, necrosis, where the nanotetrac is a very good candidate for inhibiting the cancer growth. Similarly, in the pancreatic cancer, which is one of our other objective to use this nano formulation, you can see in the left hand side control, and this is a pancreatic mpanc 96 luciferase express tumor xenograft in the left side. So you can see in control lots of luminescences coming out from the cell, but when you treat the mice with this tumor, the luminescence diminishes, meaning the nanotetrac can inhibit the growth of the pancreatic cancer cell in the xenograft model. Well, I just give you a flavor how you can synthesize the nanoparticles and how it is effective in many cancers, including pancreatic cancer, glioblastoma. So next step is again how to manufacture this nano formulation. Well, this is in progress now. This whole technology is in the process of transferring to a GMP facility for the manufacturing. Well, we didn't stop actually there. So we actually continue our next step, how we can use this nanotetrac or NDAT as a carrier to deliver different kind of chemotherapeutic drug. So we have actually used, or rather I would say we successfully encapsulate cisplatin, doxorubicin, paclitaxel, or temozolomide, or known as TMG, in our nanotetrac. We can use the same sort of microfluidizer to encapsulate those drugs, and we actually have successfully encapsulated those drugs in our laboratory using LM10. The advantage of this nanotetrac, which already has a tumor inhibiting properties, is that it can actually synergize the anti-cancer efficacy of the commonly used chemotherapeutic drug. So this way there is a potential that nanotetrac can be potentially used for metastatic cancer model, or even we have seen the drug resistant cancer model. We can use our formulation, and it gives a more depth, or it gives more efficacy when we encapsulate other kind of drug like cisplatin, or multiple drugs like cisplatin or doxorubicin together. So I am just going to give you, this will be my last slide, give you an idea how actually we can encapsulate, say, cisplatin in nanotetrac, and we have used bladder cancer as our model to see our efficacy of nanotetrac encapsulating cisplatin. So here you can see, well, this is a busy graph, but if you look a little bit carefully, you can see the cisplatin actually has a tremendous tumor inhibiting effect compared to the cisplatin or even the NDAT. Well, NDAT itself is inhibiting the tumor, but when you add the cisplatin, the inhibition capability actually increased drastically. And our laboratory also has the capacity to measure the amount of cisplatin in the tumor by using LC-MS. So here you can see that you can actually deliver more cisplatin when it is encapsulated in the NDAT rather than the cisplatin itself. So meaning you can actually direct your therapeutic load to the cancer side when you use our nano formulation, and by doing that you can actually increase the efficacy of the drug, or there might be a synergistic effect in between the drug and our nano formulations. Okay, so that was my last slide. So in summary, what we can say is that we have developed a method for large-scale synthesis of tetraconjugated nanoparticles. We also refer as nanotetrac or NDAT using microfluidizer. Initially, we have used LM10 microfluidizer to synthesize the nanoparticles, but we also have seen that different kind of microfluidizer model doesn't have any effect on the size or the desired nanoparticles we are looking for. So this can be very helpful for further scale up or commercialization of the nano formulation in near future. And in fact, actually, we are in the process of transferring for this technology for commercial development. Our nanoparticles, well, they are very good. Our nanoparticles can inhibit glioblastoma, the growth of the glioblastoma tumor model, pancreatic cancer tumor model, and many other tumor model what we have been working right now. And that is my summary of the synthesis and a little bit of the nanotetrac, and I will stop here. Any questions, I'll be more than happy to take. Thank you.
Thank you very much, Dhruva. We truly appreciate your time and effort in putting this webinar together. And thank you very much to everybody who took the time to attend. Please let us know if you have any questions or if there's anything we can do to help you develop your nanotechnology application.